to multiply and finally becomes many cells of that tissue. I mean, that's a lot of steps that we need to demonstrate. But we should be able to do this. And at the time, there was enough pieces to tell me that it was happening. Now, let's go through some of the stuff that has been shown over the past few years. I'm not giving you the whole picture here. I'm picking up, obviously, bits and pieces. My desired outcome is that when I'm done, you really get the concept here that we're really talking about the natural renewal system of the body. If you want more, we can stay here until 10 o'clock tonight. I'll give you tons of information. You will be bored to death with it, but you won't question it. So let me for now give you the encapsulated version, okay? When a tissue is stressed, it was demonstrated it releases a compound called GCSF, granulocyte colony stimulating factor, a very well-known compound known to stimulate the release of stem cells from the bone marrow, to increase the number of your stem cells in your body so we can extract them, freeze them, put you through radiation therapy, and after we re-inject your own stem cells so that you don't have any problem with rejection or anything like this. That's what we use, GCSF. Well, it was shown that before, if an organ goes into any kind of significant, challenging condition, that organ will, we don't know if that organ releases it or not. That's not known. What we know is that within 24 hours, in the blood, the level of GCSF increases. And about 36 hours later, GSF stimulates the mobilization, it means release, of stem cells from the bone marrow. We start to pick up the elevation in the number of stem cells in the bone marrow, in the blood. About 36, 48 hours later, the tissue that was stressed starts to release a compound called stromal-derived factor 1, SDF1. SDF1 is so far the only compound known to attract stem cells. If you put we call this a migration chamber. Let's say that's a chamber here, and I put at one end, I put stem cells. And at the other end, I put all kinds of cytokines, compounds known to attract the immune system. Interleukins, prostaglandins, eicosanoids, peanut butter, cheesecake, whatever. They don't move. If you put stromal-derived factor 1, they move directly where this compound is being released. That's how they're guided in the body. So they travel like this in the body, and when they get into an area where suddenly that compound is released, they put the brake, they stop there, they leave the blood circulation, and that's what they need to do. They need to follow that gradient of that compound and then migrate out of the blood into the tissue. And so SDF1 was shown to do that. In the tissue, they need to be able to duplicate, become many cells. It was shown that insulin growth factor was one of those compounds secreted by the organ where the challenge is, and then so that the cells become many cells. If you put a stem cell into an organ that is very healthy and you put insulin growth factor, nothing happens. You need the complex picture here of the stem cell moving in, the tissue being stressed, the presence of insulin growth factor. It's a beautiful picture. We could not have invented more sophisticated and more beautiful. And when they do multiply and become cells of that tissue, if this is the universal system that the body is using to renew itself and be able to regain and maintain optimal health, these cells must be able to become virtually any cell of the body. So far we're talking about brain, liver, bone, pancreatic, skin, muscle, and heart cells. And these are just virtually any cell of the body. So all that research has been done, and I'm just quoting bits and pieces of it. It is a vast literature. Just go into Medline and put stem cell and see how many thousands of hits you'll get. Okay? Now let's get into just a few studies. There are a few studies. Again, I'm picking just a few, those that really support the concept that I want you to get, which is the more stem cells in your blood, the better it is for your body. Let me show you some of the evidence that we have for this. This is a study that was done by Don Orlick at the NIH. Don Orlick asked the question, well, if there is a problem in an organ, will stem cell go there? And let me just open a parenthesis. In terms of what Orlick did. This is a phenomenon, we're discovering it today, but don't believe that it started to happen when we start to see it. It's been there for as long as mammals have existed. That's how the body regains and maintains optimal health. What is really the development here in the discovery is that now we're starting to see it. In the same way that bacteria have always existed, but we start to see it only when you develop the microscope, here's what we've discovered to see this phenomenon. One of the main development was the development of a protein that is fluorescent. It's called fluorescein. And we have developed the gene that encodes for fluorescein. If you take that gene and you put it in the nucleus of a cell, that cell will be fluorescent, but also all the daughter cells from that cell will be fluorescent. If that cell, stem cell, becomes a heart cell, you go in the heart and you will see a heart cell, but it's fluorescent. If it is not fluorescent, how can you tell that that heart cell was a stem cell before? You can't. It's an invisible process. Here's a little study that they did. Very interesting. They put a beaker and in a glass, a jar, in which they filled it with not quite water, but let's say plasma, and they put a screen in it. The screen was 0.2 microns. No cell can go through, but chemicals can go through. So they put stem cells at the top, and at the bottom they put a piece of crushed liver. Now there are ways to identify a cell. Each cell of your body have a specific marker, a signature that tells biochemically that it is a liver cell, a pancreatic cell, a lung cell, a neuron cells, a neuron. It's very easy. You raise an antibody for that protein that is specific to that cell type. You put a fluorescent tag on the antibody, and then you launch your antibody with all these cells. And if there's a liver cell somewhere, it binds to it. You go under a microscope and you see that little fluorescence attached to it. You know that that one's a liver cell. It's very simple. So they have these stem cells, the crushed liver at the bottom, and they wait an hour, 2 hours, 3 hours. They take a sample, put the antibodies, look at what we have. They're stem cells. Wait 4 hours, 6 hours, take a sample. They're stem cells. 8 hours, 10 hours, take a sample, put antibodies. They're liver cells. They no longer tag for stem cells. Within 8 to 10 hours, they have become the cell of the tissue. How it happens, nobody knows yet. But it happens all the time. You put them in the brain, it becomes a brain cell. It's very fast. If you do not have an ability to tag these cells, it's a completely invisible process. So what we have right now is a process that is just basically revealing itself to us by some of these tools. So Orlik took a bunch of mice, irradiated them to kill all the stem cells. Then he injected in them fluorescent stem cells. So it did a knot in the coronary artery, deprived oxygen in the heart, released it. There was some scar tissue in the heart, let the animal recovered, and then 2 weeks later, 68% of the affected area was now functional, filled with functional cardiomyocytes, but they were fluorescent. Okay? Now the question is, I did this by injecting stem cells that were not there originally. Am I doing something that is unnatural? So really, that's not a real thing. So here's what Orlik did. Took 2 groups of mice, did the same procedure. Ligation of the coronary artery, let the animal recover, put the animal in 2 different, put them in 2 different groups. One group was the control group, nothing was done. The other group, they injected for 5 days. This is not long-term here. 5 days, injection of cytokine, it was GCSF, the compounds we talked about before that stimulates the release of stem cells. Injection of cytokines for 5 days, okay? 27 days later, they come back and they look at the control animal. Scar tissue in the ventricular wall. There is no red. You see the enlargement here? This is all dead tissue. No new blood vessel. 17% survival in that group. Very severe signs of cardiomyopathy. When they looked at the experimental group in which they injected the cytokines for 5 days, complete renewal of the ventricular wall, filled with new, you have the enlargement here, filled with new functional blood vessels, 73% survival, virtually normal cardiac function. There's about 3,000 stem cells in a mouse. They counted 15 million new cardiomyocytes in the heart. So it tells you that one stem cell can become a lot of cells in the tissue. There are some pieces in the literature telling, at least giving us an idea. Okay, there's a real multiplication factor here. 27 days. Now, this is happening when we do an amazing, this is like you have a little bit of financial problem, and I give you like $300. I've given you here $3 billion. This is like the mega boost. When people take GCSF, you have nausea, back pain, dizziness, you don't sleep well. I mean, there's a lot of side effects to it, okay? You don't want to go that far. I'm showing this to you because at one end of the spectrum, what we have is, here's what's happening when I take the level of stem cells in your blood and I boost it to about 100-fold. Stem Enhance does 30%. It's not much compared to this. So I'm not wanting here to compare stem enhanced to this kind of situation. At the other end of the spectrum, I have the study of Tomada. Tomada greeted everybody coming for heart problems and basically took a blood sample at the hospital. He's a cardiologist. Took a blood sample and split the people in two groups. People who had no to very little stem cells and people who had a normal amount of stem cells. And then he waited 6 months, and 6 months later tested them, and the group that had the greater number of stem cells in their blood recovered much better. So at one end, we stimulate the heck out of it, and you get amazing recovery. At the other end, you stimulate nothing. You just have a little bit more naturally, and then you recover better. And that leads to a conclusion